on by Professor Vladimir V. Zarubev, who is Professor, Department of Virology, Pasteur Institute of Epidemiology and Microbiology, Russia. Amongst many positions of eminence and achievements, let me briefly touch upon few. Professor Vladimir is leading scientist in Laboratory of Experimental Virology, St. Petersburg Pasteur Institute, Russian Federation. During his early experiences, he was head of the Laboratory of Antiviral Chemotherapy and Influenza Research Institute in St. Petersburg, and also he was a Science Vice Director of Influenza Research Institution, St. Petersburg, Russia. Sir has participated in several international projects. To name few, ISTC project, laser fluorine oxygen therapy, BioLoft, improvement of influenza surveillance system in Russia, and many more. Since 2016, he works in the St. Petersburg Pasteur Institute in Laboratory of Experimental Virology, and his field of expertise in search and development of novel antivirals among synthetic and natural compounds, studying of their targets and mechanisms of activity, development of animal models of viral infections to assess actions of antiviral in vivo, and study of viruses' resistance to experimental drugs. Sir is author of 16 patents and author of more than 150 publications registered at Web of Science. Under his supervision, he has carried out 30 BSc and MSc students so far, and 7 PhD students are working or have already become PhD under his leadership. Sir, we welcome you for your talk. Before heading with this talk, I would like to introduce our chairpersons for today. Ma'am, Dr. Anita Kamra Verma. She's from KMC, Karorimal College, Associate Professor, Department of Zoology. Okay. She's Professor, Department of Zoology, Karorimal College, University of Delhi. During her early experiences, she has been a consultant, a senior scientist, and a scientist in University of Delhi. Her areas of interest includes tumor biology, type 1 diabetes, rheumatoid arthritis, immunology, and toxicology. We welcome you, ma'am. And Dr. Imtiaz Hassan, who is from Jamia Millia Islamia and one of the great bioinformaticians. Sir was ranked in top 10s on the best of bioinformaticians in India. We welcome you, ma'am and sir. Sir, we invite you for your talk. Thank you. Over to you, Professor Vladimir.
Thank you. Can you hear me? Hello. Can you hear me?
Yes, sir.
Okay. Do you see my presentation?
So,
is presentation good? Can you see my presentation?
No, not yet, sir. So you need to share your screen, your PPT.
Just a moment.
Okay.
Just a moment.
Please take your time, sir.
Okay, now you see it?
Yes, sir.
Okay. So first of all, let me thank.
Sorry, sir. You need to put that in a full screen mode. The presentation mode I'm in. Yeah.
Is that good?
Full screen. Yeah.
Now it's good?
So the last option, full screen.
Full screen, yes, it is. It's full screen. Okay, is that good?
So we request you to put it in a slideshow mode.
Yes.
Yes.
Now it's fine? Can you see the presentation?
Yes, sir, we can see the presentation, but still it is not in the slideshow mode, I think.
Tudum.
You need to, yes, you need to share the slides. I can. So please press
Yes.
the F5 button. I think that will do. Yes, on the keyboard. F5. Please press F5.
F5.
5. Yes.
Yes.
After sharing your screen, sir. After sharing your PPT first.
Okay. Что будешь делать?
Sir, you go back to your PowerPoint, share the screen, and then tick F5 from the keyboard.
Yes, just a moment. Share this. Okay. Now it's good? Ma'am?
Yes, sir.
Yes, now it's good?
F5, sir. F5. Yes, sir.
F5. Yes, I did.
Why?
Do you see it?
Pardon us. We will share it from here only.
Can you see the presentation?
Yes, we can. And
is it in a good mode?
yeah.
Okay. Shall I start?
So please continue.
Okay. First of all, dear friends, let me thank the organizing committee for inviting me here and for opportunity to present my results and to share my experience. Today, we'll be talking about antiviral drugs. And first of all, we have to understand why do we need new antivirals. Let's consider, for example, influenza virus. Here you can see the first antivirals against influenza. In fact, they are first antivirals at all. They were very effective and very satisfactory drugs against influenza. But since mid-90s, we started to observe the exponential growth of the rate of resistant strains of influenza. And now, as you can see, almost all 100% of influenza isolates are resistant to these drugs. So next generation compounds against influenza were the neuraminidase inhibitors. They were very effective too. They're very satisfactory in terms of clinical application. But similarly to rimantadine and amantadine, we observed exponential growth of resistance. And I want to attract your attention that in 2 years, the level of resistance reached about 100% in whole regions of the world. So third generation antivirals against influenza are inhibitors of polymerase. And for example, this drug, baloxavir, it's brand new. It presents on the market for only about 3 years, but nevertheless, resistant isolates have been already detected. And this is why new antivirals of alternative targets and mechanisms of activity are necessary and of great importance. Where can we find new scaffolds, new compounds from? This is a structure of drugs, antivirals against different viruses registered in last 10 years. So those violet are vaccines. They are not for treatment of viral infection, but for prevention. And among all others, only those 19 are completely synthetic. And all the rest are somehow linked to natural compounds. They are either completely natural or natural compounds inspired or natural compounds modifications. And most of them are taken from secondary plant metabolites. A couple of words about antivirals. Each antiviral should be characterized by three characteristics. This is 50% cytotoxic dose, which is the concentration killing 50% of cells. 50% inhibiting concentration, which is concentration killing 50% of virus.
Pardon me, sir. I
Sir.
cannot see the slides moving. Do you allow us to present your slides from here?
Yes. Yes, I shared the screen.
Okay, okay. Now it is visible. Thank you, sir.
Now it's good. Okay.
So,
Can you see the slide? Are they changed?
Oh.
Is it good now?
yes, sir, it is good.
Okay. So, and the third characteristic is so-called selectivity index, which is ratio of cytotoxicity to efficacy. And it's accepted generally that for prospective compounds, this index must be 10 and higher. And one more thing to be said prior to my tell. Every virus has specific life cycle, which is consisting of several stages. And these stages are very well orchestrated, and every stage takes specific time frames. So adding the compound at different stages, we can say if this stage can be inhibited with these compounds or not. Okay, now what did we do? We have several collaborating centers, which are chemical institutions in Russia and abroad, in particular in India, in Thailand, in Malaysia. We have a representative panel of viruses, very broad panel, but mostly we work with influenza viruses and picornaviruses like Coxsackie viruses, polioviruses, and so on. The first thing which I'd like to attract your attention to is plant metabolites that are strong antivirals as when they are unchanged. And first example is so-called dihydroquercetin. It's a flavonoid from larch wood. We tested this compound against Coxsackie viruses. It's virus which causes encephalitis and pancreatitis and myocarditis, a very wide range of clinical pathologies. And here you can see that dihydroquercetin is able to suppress the viral replication in mouse pancreas. And note please that its effect was even higher than that of reference compound ribavirin, which is generally accepted for treatment of antiviral infections. Moreover, the application of dihydroquercetin led to normalization of histological structure of mouse pancreas. If in control pancreas, you can see complete destruction of the tissue, but when dihydroquercetin was applied, we can observe only very moderate signs of inflammation. One more example is embeline, the alkaloid from Embella ribes. And these results have been obtained in collaboration with Professor Wiart. Hello, Christoph. And these results have been published recently. We showed that embeline.
Pardon me, sir. Sorry again, but the slides are not moving.
Not moving. Now they are?
Yes, yes, sir. Thank you.
Can I continue like this?
Sure, sir.
Okay. We showed that that embeline is able to suppress different strains of influenza viruses in in different degrees, but the spectrum of activity of embeline was very wide. We also showed that embeline suppresses the replication of influenza virus at early stages, and these led us to supposition that the target for embeline is viral protein hemagglutinin, and we showed that indeed molecule of embeline is able to bind to hemagglutinin, which is viral protein. And second group of examples is devoted to chemical modifications which confer inactive plant metabolites strong antiviral activity. And the first example of this group is so-called cytosine. It's alkaloid isolated from golden rain acacia. And cytosine itself is not active against influenza. It's non-toxic, but not active too. Its selectivity index is 2, which is very low. But when we tried to modify this molecule in different ways, we obtained a chemical library, and some of compounds from this library appeared very effective against influenza. So you can see that highlighted figures are selectivity indices of value which are 10 and more. So you can see that several compounds appeared very effective. And further, we showed that these compounds inhibits the replication of influenza virus on late stages, not at early stages like amylin D. And then we showed that the effect of citizen derivatives have been connected with influence onto the cellular cytoskeleton, which this molecule modifies the cytoskeleton in such a way that it does not suppress the transport of viral components anymore. So this is the mechanism of activity of such compounds. One more example, all you know, glyceric acid, which is effective compound of licorice, which is used, I guess, everywhere in cosmetics, in food production, and in pharmacology too. The molecule itself is not active against influenza or other viruses. So we tried to modify the molecule here and here. We covalently attached the moieties of amino acids or nitrogen bases to these two positions. And please have a look how strong these compounds became. Some of compounds has the selectivity index more than 100 and some of them more than 1,000. This is very good result, and we hope that these compounds will be tested soon in clinical trials. And one more example is camphor. All you know, camphor. This is cage compound with oxygen atom. And our colleagues from Novosibirsk Institute of Chemistry, they substituted oxygen to nitrogen. And based on this substituted camphor molecule, they created very wide chemical library of about, I think, several thousand compounds. And we tested this library against different viruses, in particular against influenza. So we showed that one of the most effective molecules was so-called camphoxin. And we selected this molecule for the experiments due to its low toxicity, high antiviral activity, and ease and low-cost synthesis. As you can see, it's very effective against different strains of influenza viruses. It suppresses the replication of virus at early stages. As you can see, the lowest virus activity was observed when the compound was added from 0 to 2 hours post-infection. Importantly, this compound appeared very effective in animal model. This is about influenza A, and this is about influenza B. And please pay your attention that in case of influenza B, camphoxin appeared even more effective than the reference compound Tamiflu, which is gold standard for influenza treatment. We also showed that application of camphoxin severely improved the histological picture of mouse lungs. If in control lungs you can see complete inflammation, at the same time the application of camphoxin led to decrease of inflammation signs in mouse lungs during the pneumonia. We also showed that camphoxin is able to directly suppress viral hemagglutinin of both influenza A and influenza B. But surprisingly, when we tried to select camphoxin resistant virus, it can be done very fastly. After 6 passages in cell culture, we obtained a completely resistant virus whose value of IC50 was about 160-fold higher than for initial virus. This is bad in terms of pharmacological prognosis. We have identified several mutations in hemagglutinin, and we explained how the mutation decreases the binding energy for camptothecin molecule. So it looks like camptothecin is not prospective due to fast selection of resistance. But we wondered if the resistant virus is of the same pathogenicity as the initial virus. So we infected mouse with equal doses of initial virus, camptothecin susceptible, and camptothecin resistant virus. And as you can see, all mouse infected with camptothecin susceptible virus died, but no single mouse died in the group of camptothecin resistant virus. These mouse even did not lose weight in the course of pneumonia. And this resistant virus did not replicate in mouse lung as efficiently as initial virus. So we conclude that the resistance can be obtained, but at the cost of pathogenicity. If virus is resistant to camptothecin, it's not pathogenic for mice, and vice versa. And in conclusion of my presentation, I'd like to say that plant secondary metabolites represent almost inexhaustible source of original chemical scaffolds. That this compound can demonstrate direct antiviral properties per se. But importantly, these scaffolds, after rational chemical modifications, even in case the initial compounds is inactive, this modification can result in development of unique effective antivirals with novel targets and alternative mechanisms of activity. And finally, I'd like to thank all of you once again for this opportunity. Thank you very much for attention. Thank you.
Thank you, sir, for your presentation. That is such a remarkable piece of work in the field of influenza viruses. So with your due permission, I would like to take up certain questions if there are any.
Sure.
Thank you. Please, if there are any questions from the audience. Okay. Thank you so much, sir.
Thank you very much too.
Moving forward, I would now like to call upon Dr. Shiv Bhardwaj. Sir is an associate scientist, Institute of Biotechnology, Czech Academy of Science, Czech Republic. Sir has gained interdisciplinary skills in biotechnology, advanced nanotechnology, and bioinformatics during the last 10 years of his teaching and research experience. At his present position, his research is focused on designing and development of protein scaffolds for both therapeutic and diagnostic applications against infectious and non-infectious diseases in humans. He is using advanced computational methods, including molecular dynamic simulation and artificial intelligence-based small molecule drug discovery pipelines for the identification of bioactive synthetic compound in disease management. He's also teaching computer-aided drug discovery techniques to graduate and research scholars. He has authored more than 50 research publications, including research articles, review articles, and book chapters. Thank you, sir. We welcome you. I would now request our chairperson, Ma'am Anita, to please present a sapling and welcome our speaker of the day. Please, sir. Yeah.
Okay, thank you. So title is evaluation of mechanistic difference in the tyrosinase inhibition by cyanidine 3-O-glucoside and catechins using computational and in vitro methods. Well, why I choose this topic? Because everyone is talking about the natural products. So we know about the tyrosinase because, okay, so in the following slide, I will explain to you. So it's not me. It's not me. Okay, neither me. So I wasn't thinking so fast. I'm sorry. So okay, let's go. Ma'am, would you please put down yeah. Okay, thank you. So let me introduce about the tyrosinase. What is tyrosinase? Okay, so no problem. So tyrosinase is enzyme. Well, like other enzymes in fungi and also in humans, we have this enzyme which is responsible for the production of melanin. And you know about the melanin, the color pigmentations, white, black, and fair colors. So, but this tyrosinase, if some kind of malfunctioning in the biochemical system, I would say molecular level, it may increase melanin synthesis, and this melanin is contributing to some skin pigmentations, or what we call it hyperpigmentations, or in neuro disease. It is also causing neurodegeneration diseases. I won't go in details into neuro. So what we have, everyone's talking about the natural product. So in tyrosinase inhibitions, we are also considering some natural products, particularly polyphenols or plant derived extracts. And why? Because we consider these compounds are safe and not toxic, and they can do the tyrosinase inhibition. So among the different compounds, among the polyphenols, we have these two compounds which are very in high concentration, up to 60% in green tea, or the C3G in black rice. Or so we have already studied, and in several literatures already reported that these compounds are tyrosinase inhibitors, but we don't know how they inhibit the tyrosinase. So meanwhile, it is another report says that if we are using polyphenols in the inhibition of tyrosinase, what happens that they may interfere with the assay, what we normally use using a spectrophotometer, because these polyphenols byproduct is similar to the substrate, that is L-tyrosine or L-dopa, and in the consecutive reactions or byproducts, they share the wavelength or absorbance, and that may interfere with our biochemical assay using spectrophotometer method. So here, if we look at the structure, the three molecules almost share the same skeleton, only the difference in the glycan or glyco groups. And these catechins or epicatechins, only the difference is in the hydrogen, like OH group, which is down the plane and up the plane, and these are isomers. So in my method, what I did, I used two methods. One is computational method. I evaluated how these compounds are interacting with the enzyme at atomic level. And then I tried to find the relationship of this computational analysis with the in vitro methods. So for computational analysis, we have a chemical databases, PubChem. I downloaded these compounds, 3D structures from there, and I collected the 3D structure of the enzyme from fungi, because we have the 3D structure for fungi, but we do not have the structure for the human. So I consider this fungi 3D structure for the computational analysis. I consider also the human tyrosinase structure from the AlphaFold, which is the database. AlphaFold database, which is AI based. Everyone knows that we are using AI these days to make the protein structure prediction. So in this particular database, they have already predicted the human tyrosinase 3D structure. So with this particular ID, I retrieve it also. And then for molecular docking analysis, I use a program, XP docking, which is XP docking protocol, which is available under the Glide program under Schrodinger's suite. And then I perform molecular dynamic simulations using academic version of Desmond, which is very user friendly, unlike Gromax or Umber. So, and for in vitro activity, we have purchased these compounds from Sigma, and then we have this enzyme also purchased from Sigma as well. And then we perform the two types of in vitro activity. One is using spectrophotometer. For that, we use a positive control, arbuthine, and then we use these flavonoids designated as with the symbols. And then we also conducted their cell toxicity using MTT assay. And then we check the mammalian inhibition assay using murine, B16F10 cell using tyrosinase enzymagraphy assay. And the total concentration of melanin production was also calculated as reported earlier. And here I'm come with the results. So before I proceed with the molecular docking analysis, and also I want to check that do I have any sequence similarity with human and the fungi. So I did the sequence alignment, and I checked that the critical residues which are essential for coordination of copper ion, because tyrosinase is a metal-based enzyme or metal enzyme, we can say, and the copper ions are required for the catalytic activity of this tyrosinase. So when I check that this, what happened then when after the alignment, I see that the critical residues which are histidine, which coordinate with the copper, were conserved between the two. And also when I superimpose the two structures from human and fungi, I see the catalytic pocket is almost same. And then, you can see here, and then this is a substrate, L-dopa. So I thought it's a perfect one, and my approach is correct. So I can continue further with the docking. So I perform the molecular docking, and I check the hydrogen bonding, and I also analyze the pose, how the orientation of the ligand is present in the binding pocket, how it is contributing or how is interacting with the metal ions, especially with the coppers there, because of copper, we know that, these are the kind of catalytic center here. So after interaction analysis, I check that what are the residues are contributing by comparison to the arbuthine, which is a positive control. And results suggested that the deprotonation of the selective flavonoids binds the catalytic residues of mushroom tyrosinase as phenolate, and presumed to follow a binding mechanism as reported for the substrate of the tyrosinase. Also, my flavonoids were noted to occupy the same plane by beading based on the structure of the chemical or the flavonoid to exhibit chrysolate-like pathway. This pathway is already elucidated to show that how these, um, the substrate or the inhibition of tyrosine happen by the known inhibitors. So I figured out that these two compounds, catechins, they follow the same pathway, same orientation in the binding pocket. So I presume that it may follow that one, unlike C3G. So ECNCH, which are catechins, are predicted to exhibit the inactivation of tyrosinase enzyme by competing with the substrate and delaying the oxidation of substrate. These were the elucidations I have predicted from the molecular interaction analysis, the poses obtained from molecular docking. So then I did the molecular trajectory analysis. I want to see that does these uh, ligands which I obtained from the docking are stable, or they have dynamic stability or not. So when I perform the MD simulation for 100 nanoseconds, I see that using, let me add to that, in academic versions, we do not have improved version of force field, but we have old version that is OPLS 2005 rather than OPLS 2004 like we have in new versions. So what I see that my protein is quite stable. The deviation is within 3 angstrom, and the same goes for my ligand also. So for example, in C3G, I have deviations mean like less than 2, but other two, for example, catechins, I have a higher deviations, but I have a stability as well. So it indicates to me that it could be that C3G is a better inhibitor, or I would say that the C3G has a better stability in the binding pocket. And as I predicted from molecular interaction analysis, it could happen that, the C3G is getting oxidized faster than the other compounds. So then I also check the interactions, how is interacting with the metal ions throughout the MD simulations. And then this interaction shows that my molecules are interacting with the copper ions. So I figured out, or my conclusion was that a C3G lacked the interactions from the catechal groups by comparison to other catechins, which may lead to rapid oxidation of our C3G compared to catechins. Other catechins are isomers, what we have, catechin and epicatechin. So then I decided, okay, let's look what happens using the spectrophotometer, because most of the people are the most of researchers even in India, they are using the spectrophotometer and they are using crude extract, and then they're saying that these polyphenols are doing inhibition of tyrosinase. And to my surprise, I see that C3G is showing more activity than the other one, which were kind of, according to literature and according to the pose analysis, it was invert. Then I decided to go for Zymo graph analysis, and then I have totally opposite results. I see that C3G is not doing any kind of inhibition, but I have epicatechin and catechins are doing inhibition in comparison to arbutin. So when I perform the zymogram analysis, my conclusions from with respect to the interaction analysis, which I perform molecular dynamic analysis and molecular docking, were supporting my results, but not the spectrophotometer method, actually results obtained from spectrophotometry. So then I decided to go for cell viability, cell-free tyrosinase inhibition assay, and melanin content analysis. So cell viability assay shows that my compounds are not very toxic, but they may show some kind of inhibitory activity at higher concentration, that is 1 milligram per ml. And then I also perform the tyrosinase-free, cell-free tyrosinase inhibition. And then I have the similar result that C3G is not doing any kind of inhibition, but the other catechins are doing. So then I also perform melanin content analysis, and I have the similar supporting results. So conclusion is this: analysis of molecular dog poses reveal the substantial role of ciliation by aiding and blockage of 3OH group by glycosylation and unconjugated seeding with catechol group in C3G compound accounts for low tyrosinase inhibition. While these observations extracted from computational analysis were in accordance with calculated in vitro enzyme inhibition, that is stronger interaction of C3G, glycosylation of 3OH group by aiding with the binuclear copper ions results in the complete oxidation by the enzymes. against other catechin compounds. Substantial interaction with the copper ions by catechol B group and 3OH hydroxyl group on unconjugated seeding of catechins play a key role in strong inhibition of tyrosinase, which is already supported by the literature. And then importantly, as results calculated are collected from spectrophory methods, but established to be not true for the selected flavonoids. It also demands improve and advanced rapid physical techniques to perform the enzyme kinetics for tyrosinase and similar enzymes with flavonoids. So takeaway message, computational analysis can be used. For example, if you have a collection of compounds, to analyze the compound, whether they will be a true inhibitor of tyrosinase or not. So I have already done this publication last year in 2019 in scientific report. And this is my conclusion. I'm going to think that computer science is no more about computers than astronomy is about telescope. Biology is about microscope. Our chemistry is about beakers and test tubes. Science is not about the tools. It is about how we use them and what we find out when we do. Thank you.
Thank you so much, sir. It is really appreciable taking out your valuable time on this Sunday morning and
Thank you,
making
ma'am.
us all aware about the enzyme tyrosinase. With your due permission, sir, I would now like to take up certain questions, if any, from the audience.
Can I ask some question? Yeah, here.
Yes, sir.
So, yes, you have done a lot of essays. You try to connect the computational biology with the experimental essays. But I find most important thing was missing. What was the rationale of selecting 3 compound? Because generally when we go for the computational study, you do a screening. Otherwise, it is very random pick and choose, or what was the rationale?
Actually, the thing was that the C3G, and we have major part was done in South Korea during my stay in Yangon University as a research professor, and then other part like computational analysis I did in Czech Republic. So why I choose only these compounds? Because we have another project running on black rice, and black rice contain like 90% C3G. So we have abundant quantity of C3G, and then we have another project running on green tea.
About your spectrophotometric assay, you are repeatedly saying you better to tell the tyrosinase inhibition assay rather than a spectrophotometric technique. And if you check the enzyme inhibition assay, what was the ICNPHH? That is
Well, actually, that's the thing. That's the thing I want to tell, because if you are using spectrophotometer method, so it's interfering with the same absorbance or same wavelength, so we cannot help it.
but it is very old enzyme. It is much, I mean,
we need to explore. That's why I said that we need some kind of improvement in the technique so that we can exactly figure out how we can do the kinetics of enzyme. You know, because if we are talking about some other synthetic inhibitor which is not showing any structural load to the substrate of this enzyme, then of course we can use this structure photometer.
No, no, my question was different. This is the well explored enzyme, so it might be having well validated assay for the kinetics. It is already well reported because it is, I think tyrosinase are used in the MSc practicals also, as I remember. So it must having, I don't know exactly because I have not worked, but it is well explored enzyme that I know. So IC50 measurements is a very common thing, and once until unless you get the very good IC50 value in enzyme inhibition assay, then you connect that your docking simulation and enzyme inhibition assay all are well connected, and then your hypothesis you are claiming it's perfect.
But sir, I'm saying you are not
So
for that.
yeah,
We
maybe.
cannot calculate IC50 for these compounds. We may calculate IC50 for other compounds.
Okay.
So in continuity, I'd like to ask you one question. In the last graph, you have shown us visual thing that the melanin content has reduced. What is the percentage of success you found? You should have, if you would have,
Well, ma'am, actually, we calculated this intensity of the band, so. we
no,
are-
You showed in a 96 well plate that melanin content is reducing. So that could have been quantitated, I'm sure.
Yeah, I have already put bar graph there.
I didn't see that. I just saw the visual. I said, no, but what was the percentage of reduction of melanin?
Well, actually, we see that in case of TCG, it wasn't much more like significant.
Mhm.
Well, in other case, we see that approximately 60% because we do need to concentrate on
Right, right.
approximately 60%, and another was approximately 40%. So,
Okay.
I mean,
Tanong mo nga, huwag mong pakialaman pera ko dyan. Tanong mo muna kay Kuya Miguel. Ay,
Okay. Anyway, thank you. Thank you and all the best.
Thank you so much, sir, for answering to all the questions so patiently. Now I would request our chairperson, Dr. Imtiaz Hassan, to kindly present a memento as a token of appreciation to Dr. Shiv Bhardwaj. Thank you. Yes, thank you, sir. Ma'am, please provide the cake. Moving forward and taking this scientific session further, I would now like to take this opportunity of introducing our next speaker of the day, Dr. Subha Basu, who has joined us online. Ma'am is a professor, genetics and molecular biology, Institute of Biological Sciences, Faculty of Science, University of Malaya, Malaysia. She is currently head of animal genetics and genome evolutionary biology lab. She has 22 years of experience serving as a teacher and researcher, and she has supervised 25 PhD students till now. She has got over 100 ISI publications with good H index. She is a recipient of several awards in the field of molecular biology and also a part of National Framework on Biosafety and Biosecurity Committee. With this brief CV and without further ado, I would now like to call on ma'am to please deliver her talk. Thank you. So
I hope you.
we are playing
Hi.
this video, and with this video, we will hear to her talk. Thank you.
Good morning to all the participants.
A very good morning to to all the participants of ICNPH conference, and my gratitude goes to my friends who like Prof. Dr. Virano, Dr. Chandra, and of course the organizing committee, Dr. Sunil. I'm so sorry that I could not come because it's during the semester break. And I hope that all of you all who's there is having a wonderful meeting and of course a good discussion. Now, today my topic is actually using of herbal intervention on gut health maintenance with genomic strategies using shrimp model. As you know, shrimps are a species that is being cultured around the world and is considered one of the most aquaculture species that is being consumed by people. We started off this research concentrating on the disease that was affecting the shrimp, which is the acute hepatopancreas necrosis virus disease, not caused by bacteria, which is Vibrio parahemolyticus. Now, if you look at what is happening globally in aquaculture, the infection started off in 2009 in China, and it has gone around the world. At the moment, intervention strategies are being investigated by a lot of researchers. Now, why shrimps? Shrimps is an organism, a biological model that I use to actually study the innate immune response and also the gut mucosal immunity. And if you look at AHPND, it's a bacterial infection that affects the gut. And there's a difference between a healthy shrimp and an AHPND infected shrimp. So as you see, the bacteria now causes an infection because of the two toxins, which is called peer A and peer B toxins. And they have affected, they produce a toxin that actually affects the mucosal region of the gut of the shrimp. Now, at the moment, there's no therapeutic option available to control AHPND. So in this case, we notice that globally in the aquaculture setting, that these strains of microorganisms have developed resistance to antibiotics like tetracycline and so on. So in a farming setting, this is a big challenge. Of course, now you can see a move to use herbal products, all right? Because we are looking at intervention that doesn't cost so much of money, all right? So today we would like to, our group would like to discuss about a traditional medicine that has been playing a role in health services around the globe. It's called Sausura Lapa. It's a member of Astraceae family. It has been heavily investigated for almost 2 decades, right? Now, S. lapa is indigenous to India, Pakistan, and China. It grows in the Himalaya region at a 2,500 to 3,500 meter altitude, and is a perennial herb. And the root is stout, about 60 centimeters, and having a strong characteristics odor. Now, Sarsura costus, a well-known medicine plant, has been actually used in Chinese herbal medicine, traditional Tibetan formula. It is also used in Arabic medicinal, and is also used in the Indian system of medicine, Ayurveda, Siddha, and Unani, and also used in Korea. So this is a work of Hassan, my PhD student, and he actually proposed his research by saying that he is Arabic. So he proposed that he feels that this herbal medicine can be the solution for the global problem that is happening in regards to infection. All right. So active constituents of S. lapa is all this, where you have volatile oils, glycosides, alkaloids, lactones, and so on. And the pharmacology of this herbal medicine It's anti-cancer, anti-inflammatory, and so on, gastroprotective, and so on. So the people in Arab actually takes this as one of their vitamins. So if you look at the demonstration of research being done for this herbal from 2005 to 2019, a lot of research has been done where it showed starting from strong inhibition against H. pylori and went on to look at it has strong inhibition to other bacteria like Streptococcus mutans and others. So we thought if this is an established herbal plant that has been investigated by many countries, we wanted to propose this hypothesis whether it can actually work on the problem that was in our lab. So the problem statement is AHPND is increasing. It is a major problem in the shrimp industry. It has caused economic losses for a long period, and drugs and chemicals are not effective against Vibrio, and you can see that they are becoming resistant. So we feel that medicinal herbs is the intervention that is actually have to be looked at in terms of it provides a solution in a very fast manner. So to assess our main aims and objective is to assess the antibacterial activity of S-LAPA against Vibrio, to obtain potent antibacterial fractionation from the roots of S-LAPA, to identify the active compounds and against Vibrio, and to analyze the interaction of retinol, which is the major constitution that we found, through interaction with peer A and peer B toxins using bioinformatic tools, and understand the mechanism of binding, and to understand the role of retinol on regeneration of hepatopancreas affected by AHPND. So this is the overview of the methodology where we have got the crude extract, we went through fractionation, and then we screen the antibacterial activity of the fractions against Vibrio, and we went on to do genomics where we did bioinformatics and protein docking and so on. This is how we prepared the samples. As you can see, I think everybody in this conference are well versed in the fact that we prepared and went through the chemical analysis, and we went through fractionation. We used GC, MS, and MS. And we actually got the roots from a local market, which is a dried plant samples. And we went through the procedure, as you can see, GC, MS, MS analysis, and this is our GC, MS in Malaysia. And the first, what we wanted to do is we wanted to determine the minimum inhibitory concentration using the crude extract first, because we didn't know whether it really worked or not. Of course, publication has mentioned it, but we wanted to test it. In collaboration with Associate Professor Dr. Chandra, which is my co-researcher, we tested against gram-positive and gram-negative bacteria. And from there, we went on also to look at the Vibrio bacteria in the shrimp itself. So we did the antibacterial test using micro plates, and we followed this methodology, right? And the methodology starts with MIC, then we looked at bacterial concentration, and we look at the percentage of inhibition. So we noticed that it has potential of inhibitory, and this was our first confidence that we felt that this herbal extract is good. And we tested it against 3 different bacteria, which was clinical related bacteria, which is under the care of Dr. Chandra. And from there, we noticed that it has inhibition potential. Then we also did different concentration, and we looked at the growth inhibition, and we were very confident, and we managed to publish this work in the sense that we established the role of S-LAPA in this research. As you can see that the fractions worked. Then we went on to test against the bacteria in the shrimp, which is against the Vibrio parahemolytic concentration, and it also worked in terms of inhibition and growth. But we went on to look at what could be the compounds related to the actual compound that is present in the extract. We started looking at which fraction that is the best fraction. The fraction that we used was ethyl acetate, all right? And we started looking at the different fractions, and we found that ethyl acetate was the one that we went on. And from the ethyl acetate fraction, we did GC-MS analysis, and we found that the highest concentration here is retinol. Okay, retinol was the compound that we picked our focus, because we felt that if you look at the results of the GC-MS analysis, the compound that was the most visible and prominent was the retinol. So we now understand that retinol is the major component of the ethyl acid fraction, and we went on to look at the postulate. Now we know that the compound retinol, which is in the literature over the other compounds, has antibacterial, antioxidant, and it has been used in cosmetic products, right? And we went on to do a further study where we compared the killing kinetics of using retinol in comparison to tetracycline, which is the antibiotic, and of course the control. As you can see, the bacteria did not die in the control, and the antibiotics tetracycline only managed to partially kill the bacteria, but retinol, which is the compound from this extract, managed to kill the bacteria within the 3 hours of time. That's very nice, and that's very interesting for us. So we went on to postulate the hypothesis. There's a group by Ng et al. In 2018 has also established the function of the retinol signaling pathway, the RHO pathway, and how it actually gets expressed in a bacterial infection. So what we did is we had a shrimp infected, which is the hepatopancreas and stomach. We understood from previous literature the toxins of peer A and peer B and bacteria actually migrates to the hepatopancreas through the intercellular gaps. So that's the pathway they actually enter to the hepatopancreas. And what it does when it comes into the stomach, at the stomach, it disrupts the tight junctions between the stomach epithelial cells, all right, allowing the formation of intracellular gaps. So in this case, there's already a previous association of that there is RHO pathway involved. So what we did is we looked at the docking mechanism between retinol and peer A and peer B toxins, and we showed that there is interaction through the docking using bioinformatics, and we actually established that mechanism that retinol is able to interact with the bacteria, right? And then we looked at the shrimps when the shrimp was infected, and we wanted to see whether the retinol gene was expressed. And in the infection, we can see that the retinol gene was expressed at a 3-hour time point and a 12-hour time point. So we are now trying to fit the puzzle, and we are trying to postulate a hypothesis where we can see whether there's an association of how to intervene the whole bacterial infection in any organisms, including human, especially in the first line of defense. We noticed that an enzyme, trans-retinol reductase, which is in the retinol pathway, it reduced at a 3-hour time point. So the mechanisms of the pathway, we are now writing the paper, and we will further elaborate, and hopefully by next year we will be able to publish, where we notice that this key element where it involves our own system, whether it's shrimp or whether it's human, you can intervene using a particular pathway to reduce the infection. So because of the presentation is very short, what we did is we have used an existing herbal compound, right, which is widely used by people in India, Pakistan, Arab, China. They have used this herbal compound and they've been using it to study its potential against bacterial infection. So what we did is we cut short and we took the herbal compound and we wanted to see whether it really verified the claims by the scientists, and it really showed inhibitory effects against a lot of bacteria, especially clinical samples, which Dr. Chandra is using for her work. So then we got excited. We said, okay, because in aquaculture world, I mean, people don't want to find fast intervention with not much of investment in research. So we took that hypothesis, which shows clearly it can inhibit bacteria. We linked it to Vibrio parahemolytic, and we showed it through bioinformatics that it has interaction, and through biochemical activities it inhibited the reaction. So that's good news for us, okay? And the reason we all are in the conference is we are trying to find solution to a problem that has been happening in the world. Any bacteria, they develop its own defense mechanism and forms resistance. But what we are trying to show is a shrimp, which relies on innate immune system, innate immune mechanism, has the potential to use its own pathway, innate system, which is the retinol RHO pathway. But sometimes a small organisms like this needs an input of a compound, and in this case, the compound is retinol, and retinol seems to be enough to fight against this bacteria. And if that is the strategy, so if there is any infection, using retinol, which activates the pathway where it forms, where we will say it's related to vitamin A, it provokes and it gives you idea that you can actually increase the transcription of the innate immune system to look at how to defend against the bacterial colonization. So this hypothesis that we are trying to do is you can see that we come from a very small lab. We can't do everything, but we try to link up to the recent publication in 2018, where they have shown Salmonella typhi can colonize the host mucosal immunity. So we are trying to, what we are presenting today is to use retinol as a possible solution. So wish us good luck. we are just trying. The whole mechanisms are still under discussion, and we hope to publish it next year. Thank you to the whole committee of ICNPH for giving me a chance to present today. It is far-fetched of a hypothesis, but we thought of sharing it, and hopefully we are able to convince the scientific committee. And thank you. Thank you, Dr. Viranud, for getting all of us together. And thank you to Dr. Sunil. And I see some of my friends I met in Sikkim is there. It's a pleasure to work and exchange ideas in a very good conference like this. Thank you.
Thank you so much, ma'am. It was such a lucid talk.
Thank you. Thank you.
Okay.
I hope you enjoyed.
Joined us for online Q&A session. We are open for questions. If any, audience, you can take, please. So it was indeed a very elusive talk. So there are no questions as such, ma'am. Thank you so much. Moving forward,
Thank you. Thank you.
another speaker who has joined us online is Dr. Bidita Khandelwal. Let me take the honor of introducing the speaker. Professor Bidita Khandelwal, Director, Department of Medicine, Sikkim Manipal Institute of Medical Sciences, Sikkim Manipal University, India. Among many positions of eminence and achievements, let me briefly touch upon few. Ma'am is an editorial member board and peer reviewer of various index journals, including the Lancet Regional Health Southeast Asia Journal. Ma'am is in national and international indexed journals in the committee of the journals. She is the principal investigator of several multicentric trials, life member of various organizations. She is a member of International Forum of Teachers of the UNESCO Chair in Bioethics. She is also a member of Research Advisory Committee for several universities and resource faculty in various national and international conferences. She is a subject expert for UPSC and a member of Governing Council in Sikkim Manipal University. Without further ado, I would like to call upon ma'am for her talk. Thank you. Over to you, ma'am.
Thank you. Please allow me to share the screen.
Yes, ma'am. Please share, ma'am. There's a green.
Yeah, I know it, but you have to allow me to. Yeah, okay.
Thank you.
Is my slide visible?
Yes, ma'am.
Okay.
Please put it in.
Yeah. Okay.
Thank you, ma'am.
So thank you very much for those kind words of introduction, and a very hearty good morning to all those who have joined today physically as well as virtually. Now, to our young delegates who are there today on this Sunday morning, they might be thinking, what is a clinician doing amongst us? And a second question, which might have also cropped up, is such a generic topic, natural products and lifestyle diseases. Last 2 days, and just before my talk, Dr. Shubha's talk, we've been talking at cellular level, molecular docking, genes, and how do you actually interact with those genes and all that. So coming to the first question, what is a clinician doing amongst us? Well, she's been trained and she has been asked to prescribe allopathic medicine. Will she actually do justice to natural products? Well, I will leave that question to be answered by the august audience at the end of my talk. The second question, why this generic topic of natural products and lifestyle diseases? You should always remember how much also of XYZ we reach. It is essential that we remember our ABC. If our foundation of knowledge is not strong, if the very small nuances of natural products is not there, or if we don't know where we want to reach, it is finally my patient. I have my diabetic patient. He is least bothered how you are extracting from a natural product. What are you doing? What are the challenges you are facing? What he's bothered is whether, apart from the various allopathic medicine that he's taking, will the natural product help him to control his sugar? And that is why we chose this topic to speak on natural products and lifestyle medication. That is, and we will discuss this topic over the following headings. We'll have a brief background check. We'll define both the terms NP and LSDs. We'll see the history, advantages and benefits and challenges which faces the natural products, brief about prevention of lifestyle diseases and role of natural products in it, and finally conclude. So the lifestyle diseases have a mortality of 41 million people per year, which is a whooping 74% of all deaths globally. And what is unfortunate is that 29% of these deaths are in people under the age of 60. Though LSD affects every corner of the world, 77% of the mortality are in the low and middle income countries. If you see, the major burden is by the cardiovascular diseases, followed by the cancer, chronic respiratory diseases, and diabetes. And these four together amount to 80% of all the premature deaths which we see. There is a public health crisis which the world is facing today. There's a good news. The infectious disease and communicable diseases are steadily decreasing. But then there is a bad news too. The non-communicable and the lifestyle diseases are steadily increasing. And so there has been a shift in the burden. And now what we need to do is find more interventions to take care of the high burden of mortality and morbidity, which is associated with the non-communicable, that is the lifestyle diseases. Over the last 2 days, you have seen that natural products are known to regulate the various pathological processes which are linked to human diseases. Hence, we have a very promising potential which is available to us in the natural product in order to treat the diseases which have a multi-stage pathogenesis. And here I would like to inform my audience that most of the lifestyle diseases have a multi-stage pathogenesis. Hence, it is of paramount interest that we understand that the need of the hour is innovative therapeutic approaches and applications of natural products. Now, before we understand how a natural product can actually benefit a lifestyle disease, very briefly, making you understand what lifestyle disease actually is. I'm sure many of you must have known about it, but then because I'm aware of the audience which we have today, I thought I'll just briefly touch on it. They're also known as longevity diseases, and they are referred to diseases where the incidence is principally linked to the daily living habits of people. So it is how we live that our lifestyle diseases occur. The main contributing factors are an unhealthy lifestyle. Now, that unhealthy lifestyle can be a tobacco use, it can be smoking of tobacco, chewing it, sniffing it, it can be a sedentary lifestyle, harmful use of alcohol, unhealthy diet, everybody into fast food these days, the junk foods, and a disturbed biological clock. And we have our phone and our mobile and our iPads being much responsible for it, sleeping at 2 a.m., getting up at 10 a.m., and having a very disturbed sleep cycle, which not only disturbs the sleep cycle, it disturbs the whole biological clock. There's another way of looking at the definition of LSD. The lifestyle of a person is a cumulative product of her physical capacity, which is coordinated with the psychological functioning. Now, this involves a pure psychological and innate control. Please note, it's a psychological and an innate control which we should have over our physical and sensory activities. And the day there is a disturbance in this initiation or control or coordination, it leads to the development and progression of various lifestyle diseases. I have already mentioned these are the 4 main types of lifestyle diseases, but there are many other which affects many other organs of our body. And to name few of them, we have hypertension, we have our statins level, that is dyslipidemia, where cholesterol, triglycerides are very high. Obesity, this is another pandemic which is now coming, most of us being on the overweight or obese side, metabolic syndrome, rheumatoid arthritis, cerebrovascular diseases, osteopenia, osteoporosis, degenerative disc diseases, sarcopenia and frailty, depression, cognitive impairment. So a psychological component is also there, and neurodegenerative diseases. There are certain characteristics of lifestyle diseases, and this is where the natural products has an edge over the allopathic treatment. These are having a very complex etiology, and they are driven by seemingly unrelated causes. It's not always a cause-effect relation that you will get. So there can be something like a rapid unplanned urbanization. There is a globalization of unhealthy lifestyle, everybody going into a different type of a lifestyle because of the generation which is there, because of the fast life which we are leading today. Everything should be like a 2 minutes Maggie and should be ready and there on our table, and the aging population. So there's a complex etiology of LSD, and there are multiple risk factors. And these risk factors may be either modifiable or non-modifiable behavioral risk factor, or they can be metabolic risk factors. Along with it, they have a very long latency period, and this stretches from years to decades. They are non-communicable. They have such a prolonged course of illness that very often you find your patient actually opting out of the treatment or developing a lot of side effects of the drugs. They just decide that no, they just don't want to continue further. And of course, there is functional impairment and disability, which makes it difficult for a person to live a normal and an active life. Now, coming to the definition of natural products, it has been observed that there is a variation in the definition among the scientific community as compared to the medicinal community. But the one which I found most acceptable is by the Food and Drug Administration, which defines natural product as any one of the following. That is, unregulated organisms or natural materials, FDA regulated unmodified natural materials or compounds, naturally occurring compound that has been chemically modified, that is the semi-synthetic one, or a purely synthetic medicinal compound which has been inspired by a natural compound. So any of these fall under the definition of natural product. So going back to the history, the benefits of application of natural product in various diseases, it includes also the infectious diseases as well as the lifestyle diseases. It dates back to the Vedic times. You can find the archives, you can see the mention of it in many of the historical things, including our very strong Indian Ayurvedic system. So then what happened? Did the journey stop there? Well, no, it continued. And today, in a very compressed period of time, we've had so much of growing evidence and confidence on natural product, its benefit, and its application. What history said was mostly not backed by scientific evidence. It was more like that, yes, this helps, this is used, and people also took it and benefited. But now we have a lot of evidence, and which has made us confident in using natural products for diseases. Between 1981 to 2010, 1,073 new chemical entities were approved by FDA. And it will be heartening for you to know that 64% of them were related to natural products. It was only 36 which were purely synthetic. Now, these were synthetic molecules containing pharmacophores, which were directly from natural products, or they were modeled on a natural product, or they were derived or inspired from nature. To add to it, in the last 2 decades, we've had an increasing number of publications which have proved that at least in vitro and in animal trials, that natural products can have a great role to play. This reflects the renewed interest of the whole scientific community to explore the great potential which lies in this area. In the next 2 slides, I just want you to see the array, the wide range which is there. So you have neurological diseases, you have bowel diseases where we have natural products acting. You have various types of carcinomas in prevention as well as management and stopping metastasis or reducing metastasis. Then you have them acting at the level of the cell, at the level of the molecule. There is a talk on diabetes, and here you see at the crux of the problem, that is at the pancreatic islets, or whether when the diabetes has not even set in at the pre-diabetic level, or in a person who already has diabetes, to see the antioxidant and the anti-hyperglycemic activities. In all these, natural products have been researched and evidenced. So what is a very major advantage which the natural product has over all other materials or products which are available? Now, since time immemorial, traditional use of NP has been there worldwide. We have had traditional herbal healers, we have traditional doctors, we have these herbal medicines going on. So there is availability of an enormous amount of ethnopharmacological information. And now on this information, we just have to piggyback. We have to leverage upon it for NP-based drug discovery. Another important advantage is that it may act as an individual and distinct entity, or it may act synergistically with other products. So these are the 2 advantages and major advantages which have translated into benefits, and anti-inflammatory, antioxidant, anti-diabetic, anti-neoplastic, anti-metastasis, and immunomodulatory effects are some. There are many more of natural products which have been evidenced beyond doubt. So we have a lot of in vitro and animal studies which have shown it. Also, it has been seen that they may act singly as an individual. So there is reduced need for combination therapy, and the toxic effect is less. I repeat, the toxic effect is less. Many a times it is felt or thought that the natural products would not have any side effect. In order to just give a brief or to make you realize what I'm trying to say as a clinician, I would take this example of green tea. People said during COVID time, Dolo became the best breakfast. I would say in the recent years, green tea has become the most in thing. There have been health benefits of consuming green tea and their isolated green tea constituents. And these benefits have been shown, including in prevention, be it of cancer or cardiovascular disease. The benefits have also been shown of green tea as an anti-inflammatory, as anti-arthritic, antibacterial, anti-angiogenic, antioxidative, antiviral, neuroprotective, cholesterol lowering, and immunomodulatory effect. Now to add to it, we have Dr. Google. We also have our WhatsApp University, and we also have our neighbor telling, I had this and this is what I have had. I have green tea for last 6 months. I have lost 6 kgs. So what has happened is people have gone into drinking too much of green tea. What we need to remember, it is beneficial up to a certain dose, and higher doses can cause serious health concerns. Also, what is alarming is that we are not even knowing all the adverse effects which may occur. You are aware of it, that green tea, the main thing is the catechins and the caffeine which it has. So it has all those side effects, and the most drastic side effect is acute liver failure. Also, you should remember that there is inter-individual variation. So effect in one person may not be similar in the other person. Now we have abundance of natural product with us. All around us, we have a lot of it. We also have a lot of data available. So what is stopping us from using natural products for lifestyle diseases? Why not we just go ahead with it? No, it isn't so easy. There are certain challenges. And the first one is to extract the active beneficial component from the natural product in vitro. Now, once you have extracted it, the next challenge which is faced is to establish its beneficial effect. Also, you will see that there is uncertainty of the beneficial effect. The constituents, due to several factors like the geographical location of the cultivation, the seasonal variation, and the method used, it affects the result. It affects the effect of that particular extract. So some herb or some plant extract, which is very good when used in Sikkim, extracted from that growing in Sikkim, might not have the same effect when cultivated at Delhi, or for that reason cultivated at Malaysia or Thailand. The next challenge which we have, that we have already proved its effectiveness in vitro. Now we need to replicate this effectiveness in vivo. You are much more knowledgeable than me as far as pharmacokinetic effects are concerned, and you know how the bioavailability in the tissue level is affected because of the pharmacokinetics. In natural product, in many of them, even now, we have lack of pharmacokinetic optimization or compatibility. So the tissue bioavailability of the tested entity is going to be not very easy. Now you found out a product, an extract, you have proved in vivo also that it acts. Now you have decided to use it as an oral tablet or an oral syrup. Now you have this gastrointestinal barrier which is there. How do you take care of this absorption? Because you need to see that the active ingredient and the amount of it which is reaching the systemic circulation is actually the desired level. So these are the challenges which we face at the lab level. There is another challenge which all healthcare professionals should be aware of. Our patient, when he will come to me as a clinician, he will not tell me about the traditional herbal medicines that he's taking. And similarly, when he will go to a traditional herbal healer or a traditional folk healer, he will not even mention the other conventional drugs that he's taking. But both of them have a dualistic effect on each other. And so we should be careful about it and should make it a point to specifically ask if you are on any other medicine. And many times they think that it is not even worth mentioning. If they're taking some natural product, they think why they should mention about it. Now, coming to the prevention of LSD, the prevention can be at the level of individual when you can have a lifestyle management. And this is where, again, the natural products have a great role to play. The societal, we need to make people aware. National health policies have to be made. And of course, global various strategies have to be done. So for prevention, we need elements with target actions, multi-sectoral partnership, knowledge and information management, and innovations. So for the multi-sectoral partnership, conferences like this, where people from different parts of the world are coming together, is the one where we should actually talk about it. What do the people want? What does the clinician want? What does the person in the lab wants? So we give our information to each other and we come out with innovations. Of course, the most effective preventive strategy would still remain the lifestyle management. So now we know that there's abundance of natural products all around us. We need to explore this potential of natural products to prevent and treat lifestyle diseases. An important barrier is the lack of public awareness. You need to make the public aware that there is a link between healthy lifestyle and lifestyle disease, and that natural products can have benefits in it. So what is the future for us? The recent investigations by scientists have been centered on understanding the molecular signaling pathways that regulate the initiation and progression of disease. It becomes important to adopt the in vitro methods and techniques which will help us to explore potential molecules and thus generate robust proofs of concept against the various lifestyle diseases. We already have a lot of positive results from natural products. Now we need translational research. So the things which have been proved in vitro, we need to prove them in vivo, and the benefits which have been proved in animal trials need to be replicated in human trials. So ladies and gentlemen, to conclude my topic, the widespread healing properties of plant-based product is well archived in our ancient scripture and also in the evolving scientific literature of the present ecosystem. Now, this is a boon. You have technology-enabled transdisciplinary research and scientific advances. And we need to use this particular technology to have the promising avenues which is available to us. And then there's no stopping in making natural product as one of the most impact-oriented, successful therapeutic destination for potential drug discovery for lifestyle diseases and tackling the growing menace. We are running out of time. We are running out of molecules. Lately, there have been many new drugs which are coming up for lifestyle disease management, but for prevention, nobody's talking about it. So this is where the natural products can play a big role. But always remember, it has to be the right product, the right extract in the right form where it is to be used and in the right quantity. So with that, I would personally, as an individual, like to thank Dr. Sunil for including me in this August conference. And on part of SMU, I would like to convey my thanks to each one of you for being there. Being a co-host, I'm very sorry that I wasn't there. I'm missing the opportunity of meeting all of you personally and interacting with you. So thank you for your patient listening.
Thank you so much, ma'am. Indeed, a very big thank you for presenting on a topic which is much required in today's post-pandemic times. Now we are open for Q&A session, if any. Ma'am, with your due permission, we can.
Yes, surely. If there's anything, and if I can answer, I would.
Ma'am, there's one question.
Yeah.
Hello, ma'am.
Hello.
Understood that you are a clinician. So I just wanted to ask you, have you prescribed any natural product-based medicine to your clients?
Okay, ma'am. So I am regulated by our National Medical Council regulations, where I am allowed to prescribe only allopathic medicine and not prescribe any other. But to
okay,
add,
so offhand for the audience over here,
yes,
could you tell us?
Yes, that is what I'm saying. Now,
Yeah.
Sikkim, I don't know how many of you have visited Sikkim, but those of you who have, we have a very rich natural flora here. Okay, so we have a lot of traditional folk healers. So many of our patients of diabetes, hypertension, gastritis, arthritis are on natural products. So and they benefit with it. So we never ask them to stop it. We never ask them to stop it. They continue with our medications.
Yeah,
Yes.
could you just name a few of them, which are available in Delhi too?
Okay, ma'am. No, I'm sorry. I will not be able to say that for simple reason. Not that it's confidential. For the simple reason that I don't know of the names. I know the local names which they use here, which might not actually work there. But again, I'll tell you, there were 3-4 patients who would come with gastritis, and then suddenly they all presented with some cardiac arrhythmia. That means their heart rates were irregular. Now, all of there's a plant known as Sisnu. That's what we call it in the local language. I don't know the biological or the technical name of it. So all of them gave this history that they had Sisnu for the gastritis. Now, the problem was that none of them had any specific amount of Sisnu which they had. They used to mix it with other green leafy vegetables and cook it. Some would make it into soup and have it, because we actually wanted to find out if this Sisnu can actually cause irregular heart rates or cardiac arrhythmias. All those cardiac arrhythmias reverted. None of them were life-threatening or none of them caused any major problem, but it causes. So in long term, it may cause. Like for green tea. Now, I was expecting one question, but it did not come because I asked this question when I attend conferences. How much of green tea can I have in a day? Because I also have green tea. I am also a fan of green tea. So how much I can have?
No, my question was, can we have green tea early in the morning?
No,
because we
ma'am.
were told we shouldn't have it
No,
empty stomach.
no, ma'am. There are 4 times where 4 periods of the day when you should not have green tea. First is empty stomach early in the morning, for the simple reason that the caffeine which is there in it will give rise to increased acid secretion and will cause problem. Second is before bedtime. Please do not take it after 7 or 8 p.m. because the way caffeine acts and makes your sleep go away, it will also do. 2 more times when you should not have green tea, immediately after your food. Immediately after your food, if you take green tea, the catechins which are there in it, they interact and they retard the absorption of the nutrients present in us. And do not take green tea when you are taking any other medication, because as of now, we are still not sure that what all effects the green tea is having. So if you're taking some other medicine, just immediately after that, wait for half an hour prior or after to that before taking green tea.
Okay, ma'am. Can I just ask one more question? You said that by changing the lifestyle, we may be able to modify this diabetes, but changing lifestyle, it's just 2 words here. So in what way do we change? Like we don't take any pizza and burger and all those things, and we don't take any outside food. We just eat home food, home cooked food. In that also we've been eating what our forefathers ate. So, but then still the problem is there.
Okay, ma'am.
So what do we do?
Why I didn't touch on that topic, because I thought my audience was different. I am so glad that you're asking me a question which actually is close to the heart of a clinician. So, so diet. Okay, so you are not into junk food. So one aspect of your lifestyle that is unhealthy diet is maybe not there. That is one. But ma'am, you would realize that your grandfathers, they took food which was very organically manufactured. There were hardly any fertilizers and all that being used. Now, even when you think you are taking a very healthy food, you're not actually sure what you are taking. First. Second, the lifestyle regarding physical activity. I hope you are on regular physical, but things have changed. Earlier, people would sit on the ground and work. They would not use a mixie. They would get up and get down. Our parents, our grandparents had done that. Now we are always on a table and a chair. We are hardly, forget about a regular 30 minutes of walk. Apart from that, our daily life doesn't have exercise. And the killer is the stress. How much also you want to remove it, the stress management has to be very, very important in order to take care of the lifestyle management. And of course, I'm sure you're not into any tobacco chewing or smoking or alcohol. So
nothing.
Yes, that is there.
Okay, ma'am. Thank you so much.
Thank you so much, ma'am.
Thank you so much, ma'am, for such a wonderful presentation, which is so
Thank
needed.
you.
Thank you so much. We have in queue our next speaker for today. And it gives me immense pleasure to introduce the next speaker, who has been a great academician and researcher, Dr. Bhumika M. Patel. She's working as an associate professor at National Forensic Sciences University and has more than 16 years of teaching and research experience. She has a distinction, which I definitely will be mentioning over here, of being the first rank holder for 6 consecutive years of B Pharma and M Pharma at Gujarat University. She was awarded Professor M.L. Khurana Medal of Indian Pharmaceutical Association. She is also the recipient of N.S. Dhalla Young Scientist Award and prestigious Rajni Bai Best Thesis Award at national level in 2013. In all, she is a recipient of more than 39 awards. Her name has been listed in the world's top 2% scientists published by Stanford University for 2019 and 2020. She has received grants for 11 research projects as investigator or co-investigator worth more than 1.6 crores. She has published more than 80 full-length papers in national and international journals. Additionally, she has published 7 books and 11 book chapters. Her areas of interest include metabolic abnormalities in cancer and cardiovascular diseases. Well, with this brief introduction of ma'am, I would now like to quickly ask ma'am to please deliver her talk.
So good afternoon, everyone. I hope I am audible.
Ma'am, you are audible.
Yeah, thank you so much for your kind words of introduction. And I'd like to first apologize that I couldn't come in person because of my new association, because I've changed recently my job to National Forensic Sciences University. Otherwise, I would have loved to come in person and attend this wonderful treat of knowledge which is being delivered. So at the outset, I would first like to thank the organizers for giving me this opportunity to be a part of this conference and present some of the work which I've been carrying out in my lab. And a special thanks to Dr. Mahavir, who has been instrumental in coordinating and communicating with respect to the conference. So I hope my slides are visible. Are my slides visible? Shall I continue or?
Yes, the slides are visible.
Okay, thank you so much. So today I'm going to present the work which we have done with respect to colon cancer associated with diabetes mellitus, answers from some herbal remedy.
Pardon, ma'am, could you please share your PPT in presentation mode?
This is in presentation mode. It is not appearing in presentation mode?
No, no, ma'am, no.
Okay,
you please,
yeah,
once,
share the entire window.
yeah,
Maybe I'll do that. Just a minute. Okay, now is it coming in?
Now it is. Thank you.
Fine. Yeah. So the topic which I'm going to present today, or the work which we have done, is for colon cancer associated with diabetes mellitus, some answers from the herbal remedy. Now, before I go on with this topic, I'd like to just take a couple of minutes to introduce the new university which I have recently joined. That is National Forensic Sciences University. This is a university with an institution of national importance now, and it is the world's first and only university dedicated to forensic, behavioral, cybersecurity, digital forensics, and allied sciences. Basically, there are different schools in our university, which are School of Pharmacy, School of Forensic Science, School of Cybersecurity, Police Studies, and so on. This is one campus in Gandhinagar, and now recently we have started different campuses across India. One in New Delhi itself, it is there. It is also there in Bhopal. Pune, there is an academy, Manipur, Tripura, and Goa. So these are different campuses which are already been established. In our Gandhinagar campus itself, we have different centers, out of which, like Center for International Relations, Cyber Defense Center. We also have a Center for DNA Forensics, Center for, okay, this is missing, Center for Humanitarian, International Center for Humanitarian Forensics, Center for Excellence of Narcotic Drugs and Psychotropic Substances, and so on. So why I just introduced is because as I was looking at the conference and the people in the conference, I saw it is a mix of a lot of different disciplines which are there. And normally this field is sometimes little unexplored. People are less aware of it. So I thought, let me introduce this even for the students, that this is one of the area which is emerging. We have a lot of courses in specialized branches in, we have for MSc in forensic sciences, MPharm in forensic pharmacy, MPharm in environmental sciences, and so on. So you can further refer the website, but just to give a brief, that this is a university which is falling under now Institute of National Importance, INI, and we have been doing this. I am a pharmacologist for last 16 years. I've been in the area of pharmacology, and now I'll be dealing, I mean, in this university, in addition to pharmacology, toxicology is the area which I'll be working upon. So having that brief preamble, now let me look into my topic for today. So I said diabetic associated with colon cancer. To just begin with giving you a brief overview about what, I mean, diabetes we all know, but what is the prevalence? We all know it is highly prevalent. And the data from International Diabetes Federation, it mentions that by 2045, it is going to increase to around 700 million, which is a 51% increase. And if you look at the areas, India is dark in color, which suggests, yes, India has a lot of cases of diabetes. Similarly, if I look to colorectal cancer, of course, as compared to other parts, India has having less cases, but at the same time, a recent report which says that there have been 20% increase in number of cases of colon cancer in India as compared to uh, 5-year back data. So we all know, we just had a discussion about lifestyle, just previous session we were discussing. So lifestyles have changed, the food habits have changed, which have also increased the rate of colorectal cancers in India. Now, we know about diabetes, we know about colorectal cancer, but there is an association between diabetes and colon cancer itself. There are several reports which suggest that, you know, the patients who are at have type 2 diabetes, they are at increased risk of colorectal cancer. Around 30% increased chances of colorectal cancers are there. Also, if there is an uncontrolled diabetes with an uncontrolled or very high levels of HbA1c, that suggests as an independent predictor of clinical behavior of patients with colorectal cancer. So patients with colorectal cancer having diabetes which is uncontrolled, that is also one of the predictor for the cancer itself. Also, the histopathological features of patients with cancer, they are worse as compared to in diabetic patients as compared to non-diabetic. So if I compare diabetic versus non-diabetic, both of them suffering from colon cancer, diabetic patients will have worsening of the disease or more histopathological changes. In addition, the patients who from type 2 diabetes, they start depending, you know, off late diabetes converts into type 1, and they have to have hyperinsulinemia, and chronic insulin therapy is also given. Again, those patients are at high risk of colorectal cancer. So yes, there is an association between diabetes and colon cancer. We say colon cancer is one of the risk factor that, or you know, one of the chances is there if they have diabetes. In those patients, development of colorectal cancer is common. Now, so we thought, let me have something which we can target, which is a common target for both diabetes as well as colon cancer. And we found that one of those targets are cyclin and cyclin dependent kinases. So basically, cyclin dependent kinases, they are serine threonine proteins. Cyclins and cyclin dependent kinases, they play a major role for the cell cycle. So at every place of cell cycle from G1 to S to G2 and mitosis, these cyclin dependent kinases and cyclins, they play a role in the entire thing. There are different types of it which play a role. Cyclins, they have well conserved amino acid sequence, and which is essential for activation of cyclin dependent kinases, CDKs. And most commonly, it has been reported that CDK4 and 6, they are elevated in cancer cells. And cyclin D is also found to be overexpressed. So this is a role of cyclin and cyclin dependent kinases in colon cancer. What about diabetes? Okay, so in colon cancer specifically before that, so there is this RB protein which is associated or which is present as a complex with E2F protein. What does this CDK4 do? CDK4 causes phosphorylation of RB protein, retinoblastoma protein, and because of which there is gene transcription. The gene transcription of the colonic or the cancerous cell, that transits into enzymes and proteins, they move on into synthetic phase, and the cell cycle advances to the synthetic phase, and it keeps on multiplying. So this keeps on going on, and that leads to cancer. So that is how CDK, especially CDK4, which is upregulated in colon cancer, play a real role for cell proliferation. In addition to that, this P53 gene we already know, anti-apoptotic, it inhibits CDK4. So in colon cancer, again, P53 is reduced. Now, what about in diabetes? Now, in diabetes, it has been reported that again, CDK4 along with E2F1 and this phosphorylated retinoblastoma protein, which we saw in other earlier slide, it is responsible for insulin secretion. Basically, CDK4 again, with it acts on the complex in pancreatic cells. It causes dissociation of retinoblastoma protein from E2F, and this E2F thereby regulates potassium channels, ATP sensitive KIR 6.2 channel. These ATP sensitive potassium channels, we all very well know they are playing a major role in diabetes. Now, I'm sorry, how to, okay. So now this potassium sensitive ADP channel, they play a major role in insulin secretion. And because of this, the insulin secretion becomes faulty. So the idea is if we can inhibit CDK4, we can target both hyperinsulinemia as well as we can target colon cancer. So this is how it works with respect to diabetes and colon cancer association and role of CDK as a common thing between them. Type 2 diabetes develops into insulin resistance. Because of insulin resistance, there is increase in demand of insulin, which causes beta cell proliferation and hyperinsulinemia. This increases IGF-1. Now, IGF-1 insulin growth factor, it's a growth factor. So it is obviously responsible for multiplication. This IGF-4 or IGF-1 causes more and more secretion of IGF binding protein, IGF-BP1, that is a binding protein which binds to IGF. So because of this decreased secretion of binding proteins, free IGF is more. So again, I said IGF is a growth factor. So because of that, it causes alteration in the colonic mucosa, which causes uncontrolled growth of the cells leading to colon cancer. And we already saw role of CDK is there in the insulin secretion in hyperinsulinemia, as well as it controls the multiplication of cells, growth of cells. So we thought, why don't we target the CDK4 and see if it works in diabetic colon cancer, meaning colon cancer which is associated with diabetes mellitus. So now we went back and looked into some herbal products. We found that lignans are reported to be having CDK4 inhibitor activity as well as they are anti-cancer. Then we looked into lignans. Lignans are found in variety of plant-based foods, legumes, fruits, vegetables, etc. One report we found that one of the plant or one of the seeds, flax seeds, they have a very high amount of lignan which is present. So we looked into flax seeds. Of course, flaxseed Linum usitatissimum is its biological name, or C, linseed, flax, they are Ataceae, they are all its common name. And a lot of people take this as a mouth freshener. They take it post lunch in different formulations, or also now flax seeds are available. You have fortified breads, fortified, eggs, flaxseed oil, which is available. A lot of such products are already available in market. But when we wanted lignans. So lignans are not available in fat oil of the flaxseed. So, normally if you are extracting a flaxseed oil, you extract the oil and whatever is remaining, that is discarded. So that discarded product actually contains a lot of lignans, and that is what we targeted. So that lignan majorly which is present in that remain is the secoisolaricinol diglucoside, or in short, I say SDG. SDG is again already reported to exhibit anti-cancer, antioxidant, anti-atherosclerotic, and renoprotective effect. Anti-cancer basically for breast cancer it is reported. So why we thought let us explore this, and then thus our objective was to study. We studied two things. What I'm presenting today is sequential two studies we carried out. One is just we looked into the rich extract of SDG, and then we also studied the effect of SDG itself. So we studied both the extract as well as pure SDG, which we later on did it in next group of studies in both diabetic colon cancer, and thereby we wanted to establish its mechanism of action. So first of all, we prepared SDG lignan concentrate. I'll not go into the details. All are persons who are experts into herbal. But basically, after defatting, as I said, we remove the oil, and the remaining powder which is there, defattened powder we extracted to collect a SDG lignan concentrate, what we call it as LSC now. Also, we obtained, that was our first set of study, and then we obtained pure SDG also for our second set of studies. I mean, of course, the protocol was approved from the Institutional Animal Ethics Committee, and we used male Sprague-Dawley rats for this particular studies. And we divided into different groups, which will be clear when we'll see the results. Now, how did we induce diabetes in colon cancer? So we used standard model of both diabetes as well as colon cancer. Streptozotocin, low dose with high fat diet. It is a reported animal model for diabetes, type 2 diabetes. So we gave high fat diet for 2 weeks. Then we administered a low dose of streptozotocin from at second week. And at 6th week, we confirmed the animals by performing fasting glucose tests that all of them were diabetic. Few animals which were non-diabetic were excluded. The ones which were diabetic now were administered with DMH, which is again a carcinogen, dimethylhydrazine. It's a carcinogen. It was given twice weekly in the dose of 25 milligram per kg IP route, daily from 6th to 18th week. And then 18th week, it is reported they developed cancer. They were kept as such for progression of cancer. And 24th week, the study was terminated. The treatment we started from 6th day itself, parallelly till 24 weeks, we gave this treatment for both of them. And after this, we determined several parameters like general glycemic. We'll see all of those, which will come one by one. In addition, we also carried out certain in vitro studies like MTT, clonogenic assay, scratch wound, and cell cycle analysis using flow cytometry using HCT15 cell line, that was Annexin 5 HCSA. So now let me just put on our results. So first, to begin with the in vivo studies results. So looking into the diabetes marker, these are the results of the lignan concentrate. DCS is the lignan concentrate treated group in disease treated. And we, as I said, we carried out a second set of study using pure SDG also. So we found that I'm not presenting all the results, but yes, we found that it produced a better control in the oral glucose tolerance test. Also, HbA1c, which is a marker for diabetes for 3 months marker for diabetes. Of course, there was an increase in HbA1c in the diabetic colon cancer group. Treatment with both, you can see here, this is lignan concentrate and this is SDG. There was a significant fall in that, suggesting its role in diabetes. Similarly, hyperinsulinemia was treated. There was hyperinsulinemia in the diabetic colon control animals. The lignan concentrate as well as SDG, both of them significantly reduce the insulin levels, suggesting that it produces control in serum hyperinsulinemia. So that hypothesis of controlling insulin-like growth factor mediated colon cancer is controlled here. Then looking into tumor markers, when you look into tumor incidence, percentage tumor volume, and tumor burden, all of them were controlled with both SDG as well as SLC. I've put up the results of only SDG here. So overall number of tumor or tumor formation was controlled with the treatment. Then we looked into the cancer marker, CE, carcinoembryogenic antigen, that is a marker specifically for colon cancer. It was elevated in the disease control group. Both the lignan extract as well as pure SDG produce a significant reduction in the CEA levels, which again suggests control in the tumor. Then we also looked some non-specific markers like interleukin 1 beta and TNF alpha. Both of them are markers for because of the host tumor interaction, there is an inflammation in colon cancer. So even those were reduced by the treatment with SDG as well as SLC. Then looking into some oxidative stress marker from the colon tissue, we extracted out the colon tissue and carried out some oxidative stress analysis. MDA, which is a pro-oxidant malondialdehyde, that was elevated in disease control and again treated with SDG. The SOD, which is an antioxidant, and GSH, which is also an antioxidant, those levels were reduced in the colonic tissue. And again, they were elevated by the treatment with SDG itself. Then we moved on into the histopathology study. So if you can see this is a normal colon. There is a normal epithelial tissue with the mucosal layer is also intact here, you can see. In disease control, there are hyperplastic cell, there is epithelial damage, and also at some parts, there were completely developed tumor. But if you see here with the treatment of this is for lignan extract, there is a restoration and visible epithelial wall with restoration and prevention of tumor. And same are the results when you compare with the pure SDG. So if you see the results of pure SDG also histopathologically, they are putting a control. Then we also carried out PCNA studies, proliferative cell nuclear antigen assay. So basically, this is an PCNA is a marker for cell proliferation, and during immunohistochemistry we did this. So if you can see here, the dark brown, yellowish brown colored are disease control, which I mean those indicate positive PCNA. So more the PCNA, more is the proliferative capacity of the cell. So you can see a lot of such spots here, wherein if you see in the treated group, they are very less here. In the entire this thing, you can see very less, and upon quantification using image analysis also, we found that this is being proliferative index is reduced by a greater extent significantly by the treatment. Then, as I said, what we did is in the first set of experiment, we just did the in vivo studies and we saw that it is working. So that pure SDG, what we obtained, we subjected it to some in vitro analysis also to provide some additional confirmation to our result. So to start with MTT, which is a basic cell viability assay, we found that there was a significant increase in percentage cell inhibition at 24 hours with different doses of SDG. Then we also carried out scratch wound healing assay, which indicates about the migration capacity of the cell. So if you see, these are the control cells at 0, 12, and 48 hours. What we do is we grow the HCD15 cell lines, and then we create a scratch using a pipette. And normally at 12 and 24 hours, they'll become confluent again. You can see this particular scratch, which is there here, is not seen at 12 or 24 hours. But gradually in the treatment group with 2, 3, 5, 4, and 5 microgram, you can see they are not able to migrate, meaning that these cells lose the capacity of, or because of the treatment, there is a decrease in the cell migration and overall cell transmigration. And again, as I said, the wound closure, when we quantified it at the highest dose, we saw a dose dependent prevention of the wound closure in the treated group. Then this is a clonogenic assay. So clonogenic assay is basically colony forming capacity at different time interval. So we seed the 6-well plate with the HCT cell line at a specified concentration, and we allow it to proliferate. And at 48 hours, we stain and we see the number of colonies which are formed. So you can clearly see at 48 hours, there are a lot of colonies, and there is a dose dependent reduction in the number of colonies which are being formed. These are just the representative images. So you can see here at the quantification, a number of colonies formed are reduced. Then finally, to ascertain more about its mechanism of action, we carried out Annexin 5-HC assay using flow cytometry, FACS analysis. And what we found is, if you look at the SDG, it's like this is a population of SDG, this is of control. You can see majority of the cell are densitized in the early apoptosis and majorly late apoptosis. So SDG itself also promotes the cell to undergo apoptosis. So we could say that SDG is working with respect to prevention of cell proliferation as well as induction of apoptosis. And then finally, some few studies to determine its mechanism of action. Like before, we carried out purely with SDG. The extract, like I said, we carried out first set of experiments using the extract, and then we wanted to see for the SDG. So how did we go with that? Is what we did is since we knew SDG is a lignan extract, we carried out first the docking study with CDK. And these are the standard docking, compounds which act on CDK, purvanol, rosuvastatin, and staurosporin. And we also docked SDG. And what we found, the docking score of SDG was good. In addition to that, the number of hydrogen bonds formed by SDG were very, very high, suggesting that yes, SDG works majorly. I mean, from the in silico study, yes, it does works through CDK4. And then we finally confirmed this using mRNA expression also. And if you can see here, we found that there is a significant reduction of CDK4 mRNA expression when treated with the SDG. So basically, this was what we studied. And finally, I was given 20 minutes. Yeah, I'm on time. So I would like to conclude that I initially mentioned that this is how through CDK, CDK plays a role in colon cancer. And so we suggest that this drug, the extract as well as pure SDG, inhibits CDK4. And because of this, it also upregulates P53. I have not gone into those set of experiments because I said it induces apoptosis. We also carried out expression studies of P53. It upregulates P53 also, and thereby we suggest that it prevents colon cancer. So these are some selected references, and I'm thankful to you for your patient listening, and I'll be happy to answer any of the questions, whatever you have.
Well, thank you so much, ma'am, for such an insightful talk on colon cancer. Since we are running short of time, I'm afraid we need to skip the Q&A session.
Okay.
But with your due permission, ma'am, I would request if there are any questions be directly mailed to the speaker.
Yes.
Thank you. Okay, we will take a short break since we are running late already. So I would request you all to kindly join us at Archery Ground for a small tea break. I request everyone to come back by 12 sharp to start further. Thank you, everyone. The session is closed now, ma'am. Thank you.
Thank you. Thank you.